Hi everyone, I'm Saranya. I'm going to give a brief overview and history of forensic DNA profiling. On August 26, 1999, a female student of University of Virginia woke up after finding a gun pointed at her. The assailant sexually assaulted the victim and raped her. The male student who was spending a night with her was robbed. The assailant forced the student to have a shower to get rid of any forensic evidences, but the assailant already left a beer can with salival samples and semen sample on the bedsheet. The salival sample and the semen sample matched with each other. After 6 weeks of crime, the DNA sample of the assailant matched with one of the DNA sample in the Virginia database. The criminal justice system consists of three broad areas: law enforcement, scientific analysis, and legal proceedings. Law enforcement consists of investigators and detectives who served in police department. They submit the crime scene evidences to forensic laboratories. The forensic laboratories compare these evidences to reference samples or DNA in the databases. The scientific report is then generated. Legal proceedings depend upon the scientific report and is presented in front of the court. In this slide, I'm going to give an overview of forensic DNA typing. In this slide, I'm going to give an overview of forensic DNA typing. DNA profiling was found by Alec Jeffrey in the year 1985. DNA sequences are repeated next to each other and it varies between individuals. These repeats are known as variable number tandem repeats or VNTRs. The method to analyze VNTR is known as R-flip or restriction fragment length polymorphism.
Uses restriction
Forensic DNA
enzymes to cut
testing
the DNA
using
around
R-flip
VNTRs.
was used to catch
The
Colin
first
Pitchfork,
forensic
who
DNA
was a
testing
rapist and a
using
murderer.
R-flip markers was to catch Colin Pitchfork, who was a murderer and a rapist.
In this slide, I'm going to explain about the basic principle of forensic DNA typing. With the exception of identical twins, each individual is unique, half inherited from mother and half inherited from father. Forensic DNA test examines only a subset of human genome in order to save cost and also the time. Statistical calculations are based on random match probabilities. DNA analysis using star markers are used for parentage testing, disaster victim identification, genetic genealogy testing, and historical and missing person investigation. Parentage testing, it is used to find a child's father. Disaster victim identification is used to put back pieces together after a massive disaster. Genetic genealogy or ancestral DNA analysis is to find one's own heredity. Historical and missing person investigation helps to link the recovered bones from unknown individuals back to their own families. In this slide, I'm going to explain about the steps in DNA analysis. DNA extraction can be performed from biological material. After extraction, DNA needed to be quantified in order to know the amount of the DNA present. DNA can be amplified using polymerase chain reaction. Multiple loci can be amplified using star markers. Separation and detection of Pcr products can be performed using capillary electrophoresis in a genetic analyzer. The samples can be compared to the suspect and reference sample, and if a match occurs, DNA profile is compared to a population database. The case report can be generated using the probability of random match. DNA analysis always requires comparison between two samples, question and reference sample. After the comparison, an exclusion report, inclusion report, or inconclusion results can be obtained. After the results are obtained, a lab report is generated. If the reference sample is not present, then the question sample is compared against a computer software that have DNA database of previous offenders. K, or reference samples, comes from a known source and there is no need to determine the origin. In paternity testing, alleged father or father's DNA sample, K, is compared against the child sample. In massive disaster investigation, victim or missing person DNA will be the question sample is compared with the biological relatives. Soldier's remains may be identified through blood sample collected from the soldier when he is alive. After the comparison results, the results can be inclusion, exclusion, or inconclusive. The lab reports inclusion result as the DNA profile from sample Q is consistent with the DNA profile of sample K. Therefore, sample K cannot be eliminated as a possible contributor of genetic material isolated from the sample Q. If a comparison finds the Q and K samples equivalent or indistinguishable, then a statistical evaluation is performed by comparing the results to a population database. In this slide, I'm going to explain about the history of forensic DNA typing. In 1985, Alex Jeffreys developed multi-locus restriction fragment length polymorphism props. In 1986, DNA testing goes public with Cellmark and Lifecodes in the United States. In 1991, fluorescent star markers was first described. In 1993, first star kit was available and sex typing loci amylogenin was also developed. In 1997, core 13 star loci was defined and Y chromosome star marker was defined. In 1998, FBI launches National Combined Index System. In 2000, FBI and other labs started using multiplex stars and stopped using R-flip markers for DNA profiling. PowerPlex 16 kit enables first single amplification of coded stars. In 2002, FBI Mitochondrial DNA database was released and Y-star 20plex was published. In 2003, National Index DNA Data System in US exceeds 1 million convicted offender profiles and UK database exceeded 2 million convicted sample DNA profile. I'm going to introduce about the historical methods of DNA typing. In this slide, I'm going to give a brief comparison about the different DNA typing technologies that was used from past to present. Technologies used for performing forensic DNA analysis differs in their ability to differentiate between individuals. DNA testing earlier took 6 to 8 weeks. Now it can be performed in very few hours. Techniques varied from single locus and multi-locus probe restriction fragment length polymorphism to the more recently polymerase chain reaction-based assays. A large blood stain with well-preserved DNA was required for a good DNA profile, but now tiny amounts of sample can yield a useful DNA profile. DNA markers have been divided into 4 quadrants based on their power of discrimination and also their speed of analysis. An ABO blood group determination was the first genetic tool used for distinguishing between individuals. They can be performed in a very few minutes, but it is not very informative. It only gives information about the exclusion of an individual from the crime. Multi-locus R-flip probes are highly variable between individuals, but require a great deal of labor. Multiplex star markers are valuable because they can produce highly discriminating results from a small and degraded DNA. Mitochondrial DNA, which is shown in the quadrant with the lowest power of discrimination, takes the longest sample processing time, involving severely degraded DNA samples while associating maternally related individuals. There are instances where nuclear DNA is either so degraded or present in very small amount. In such cases, mitochondrial DNA is the only alternative for forensic DNA testing. There has been a gradual evolution in the adoption of various DNA typing technologies. Validation of new methods is crucial in maintaining the high quality results. In 1990, Karl Leinsteiner, an Austrian researcher at the University of Vienna, discovered that bloods sometimes agglutinate when they are clumped together. His work resulted Nobel Prize in the year 1930 that eventually identified four blood types: Group O, A, B, and AB. Leone Lactus, a professor at the Institute of Forensic Medicine in Italy, developed methods for typing dried blood stains with antibody tests for the ABO blood groups, and this genetic data was present in Italian court in the year 1950. Over the next few decades, the use of ABO blood typing was used for forensic cases in England and spread to the European countries and also in United States. Another blood group system, MN system, was used for differentiating few people. Alexander Vienna discovered that the RH factor while working with rhesus monkey. In this slide, I'm going to explain about forensic protein profiling. Amino acid sequences in some proteins varies among individuals in human population. Protein polymorphism in the human genome aided in protein profiling to distinguish between the reference samples and also the question sample. Human red blood cell erythrocyte as well as blood serum has a number of isoenzymes. These isoenzymes are multiple forms of protein enzyme that can catalyze the biochemical reaction even though there is slight differences in the amino acid sequences. However, there are typically 1 to 3 forms of isoenzymes, making them fairly poor at indicating the difference between people. In this table, there are few isoenzymes are given and their discrimination potential is also explained. Restriction fragment length polymorphism or RFLIP. Alec Jeffreys in 1985 found restriction fragment length polymorphism or RFLIP. In his three Nature publications, he mentioned about the repeated DNA sequences and genetic inheritance can be used to track the differentiation among people. In this process, a sample of blood or other biological material was collected. DNA was then extracted from the cell by breaking open the cell membrane and removing the protein packaging around the DNA. Next, a restriction enzyme was added to cut the DNA molecule into smaller pieces. Most commonly used restriction enzyme used in the United States was HAE3 that recognizes GG/CC. GG is guanine, CC is cytosine. Every time GGCC appears in the genome of a sample being processed, two DNA fragments will be produced, one ending in GG and the other ending in CC. The reverse would be true on the other strand. The chopped up DNA fragment were then separated by fragment size on an agarose gel electrophoresis. After this, the separated DNA fragments were transferred onto a gel, that to a nylon membrane. By using highly alkaline solution, the DNA strands were rendered single-stranded. A chemiluminescent probe which contained VNTR sequence was then allowed to hybridize the DNA attached to a nylon membrane. Excess probe was then removed with several washes of the membrane. Finally, location of a probe was noted by placing in contact with the extra film. Resulting position of a band in autoradiogram was then recorded. The band positions can be analyzed by means of molecular markers analyzed in parallel. PCR-based testing. Kary Mullis invented PCR early in 1980s. About the same time or flip, another technique that came into being, it's known as PCR, invented by Carey Mullis, and there was no polymorphic genetic markers available at the time of amplification. Pcr amplification was performed to amplify the entire VNTR region, and this is known as long-range PCR. In the late 1980s, microsatellite, better known as short tandem repeats, were being reported. And these short tandem repeats are shorter repeat units with 2 to 7 base pairs. The commercial star kit capable of multiplex amplification consisted of only 3 loci. They are CSF IPO, TPOX, and TO1, and they are known as CTT triplex. Silver stain detection method was quite effective for laboratories that want to perform DNA typing for a smaller startup cost. No expensive instrument was needed, simply a gel box for electrophoresis and some silver nitrate and other developing chemicals. Fluorescent tag PCR kit. The capability of simultaneously detecting star alleles was enabled by labeling the PCR product with different colored fluorescent dyes. Today, the multiplex PCR assays capable of 15 or more stars are routinely used in forensic laboratory to produce DNA profile. Mitochondrial DNA sequencing was developed to recover the DNA results from bones, hair, and teeth. Sanger sequencing. Mitochondrial DNA analysis involves DNA sequencing known as Sanger sequencing. The Sanger sequencing process involves incorporation of dideoxynucleotide triphosphate as chain terminators. There is no hydroxyl group at the 3' end of a DNA nucleotide, and the chain group terminates when the polymerase incorporates a ddNTP into a synthesized strand. DNA sequencing may be reliably performed on a variety of platforms using the genetic analyzers.